Welcome. Today we are going to look at the lymphoid organs. These are the organs where immune cells are produced, they develop, and these cells learn to differentiate self from non-self, i.e., they learn to identify foreign bodies which they are supposed to destroy. We'll be looking at the structure and function of these lymphoid organs. So on the structure, there are a few common things that we need to know before you can start discussing them. First, most of these organs are encapsulated. What that means is that they are enclosed by a capsule. Then internally, they may be divided into compartments known as lobules by an extension of this capsule inwards. The extension normally forms a wall known as septum in one. This septa may be called trabecular. So you check on that. Then, since these organs have living cells which are dividing, they normally have blood vascular system, which is transporting food or nutrients and oxygen to the cells and carry away developing immune cells plus waste product from the organs. So these are some of the few common things that you'll find in them. Another common thing is that they will be antigen presenting cells at the stage where these cells are supposed to be learning foreign versus self. So these antigens must be presented to them by antigen presenting cells. This will include macrophages, dendritic cells, and even epithelial cells of these organs. So let's start by looking at the main divisions of lymphoid organs. We have primary or central lymphoid organs, and then we have also secondary lymphoid organs. So we'll start by looking at the primary lymphoid organs. Before we start, let me state that in primary lymphoid organ, the major activity is production of immune cells. The other major activity is growth and development and even specialization, that's differentiation of these immune cells. So the major thing that happens in secondary lymphoid organs is continued differentiation, but the main thing is actually learning the foreign bodies, learning what is an antigen and what is self. So it is here where most of antigen presenting cells are presenting the antigen to these forming immune cells. In secondary lymphoid organs is where they also now start the active work of eliminating or destroying the antigens. So let us start by looking at primary or central lymphoid organs. As I've said, they are the center of production of these cells. But normally it is the bone marrow which produces almost all the immune cells. The thymus, which is the other primary lymphoid organ, is only specialized and allow development of T cells. So let's go through these things. They form site of production and development of and maturation of white blood cells. They are bone marrow and thymus. All the blood cells are produced in bone marrow, I said. B cells are produced in bone marrow and mature there. T cells are produced in bone marrow but mature in the thymus Throughout our discussion, we'll be looking at the development mainly of these two cells and a few antigen presenting cells that I had mentioned earlier. Then let's move on and now see the structure of the bone marrow. This diagram is the diagram along bone. It shows red bone marrow and yellow bone marrow. Yellow bone marrow stores lipids. Red bone marrow is where the blood cells are formed. Now remember, white blood cells are the immune cells. So it is this place where the activity of producing cells is. So let's go back and look at the structure. So the structure of the bone marrow, as I said, first it contains what we call stem cells or hematopoietic cells or tissue. Stem cells are cells which are capable of dividing throughout the life of an organism. Most cells specialize and stop dividing, but these cells do not specialize. In plants, they are found in the cambium. So these stem cells, apart from continual division, the cells they produce, daughter cells, can specialize or differentiate into any type of cell. They can form sperm cells, they can form bone cells, they can form nerve cells, etc. But in this case, they will be forming different types of blood cells, red blood cells and white cells. So again, we have vascular sinuses. These are the blood vessels which bring blood to the bone marrow and take them away. They are divided by trabecula, which form a meshwork when you observe through microscope. So these are the walls, I say, that divide the bone marrow into compartments. So the blood vessels bring food and nutrients and take away waste products, and they form immune cells. Let's summarize the functions of the bone marrow. It gives rise to all lymphoid cells. The V lymphocyte mature in bone marrow until lymphocyte in thymus. We looked at that. It has pluripotent stem cell. I've talked about that. It forms melanocyte for B cell production and differentiation. We have said that. Platelets, which help in blood clotting, so produced in bone marrow. Yellow marrow stores lipids. The function of the bone marrow in birds is done by bursa or fabrices. So you remember that. Then let's look at thymus. Thymus is a flat bilobed structure. It has two lobes. It is located in thoracic cavity. We'll see a diagram which show you the location of these lymphoid organs. So it will help you in learning where they are located. So it is a place where T cell lymphocytes develop and mature. So the cells in the thymus are thymocytes. These are immature T cells which are developing. Then phagocytic reticulum or reticular epithelial cells should form the antigen presenting cells. So it is capsulated. Each lobe is divided into lobule by trabecula. I had mentioned that. So each of the lobule is divided into outer cortex and inner medulla. I said that. But the one thing that we need to mention that at birth we have the thymus developing. It grows to maximum around puberty. After that, it atrophies. That means it reduces in size until it is replaced by fat, disappears somehow. So let's look at development of T cells in the thymus. What things do T cells acquire in the thymus? First of all, immune cells must have receptors on their surface for identifying the antigen. The one of T cells is called T cell receptor. Some authors just say TCR. So, as I said earlier, immunology has its own vocabulary, so know that TCR means T cell receptors. Some textbooks just write TCR in diagrams. Some other things which they acquire are T cell markers. We'll see that some cells are just referred to as CD4. In fact, this is T helper cell, CD8, T cytotoxic cell, and CD2. So remember that those are acquired also. So let's then look at what happens finally when the T cell now develops. Here, there's no activation of T cells, but T cells are exposed to antigens for one reason: to develop a T cell which can differentiate cell from non-cell, So what happens here is that antigen presenting cell, in the cortex it is just epithelial cells, thymus epithelial cells, will present antigens to these developing thymocytes. So there are thymocytes which will recognize that these are foreign substances and these are immune cells. So they will attach to foreign substances and not body cells, They will note that these are foreign substances and these are the body cells. So body cells have self and antigen and self. So they will touch non-self rather than self. So those ones which are attaching to self just as non-self cannot recognize the difference. So they die by what is apoptosis. Programs killing, they are killed. So they are eliminated. Majority of T cells, developing T cells, die like that. Only a few become clever to realize that they self and non-self, and they continue to develop. The same process occurs here, but here the antigen presenting cells are now macrophages and dendritic cells. So the mature T cells then leave thymus. There's something that, this is the diagram that I've really talked about. So you can see the capsule there surrounding the whole of the thymus. Then they extend inwards into trabecula or septum. Then you can see the outer part of each of those lobules is called cortex. The inner part is medulla. Then the green things are the thymocytes, the developing T cells. Then you can see this is a antigen presenting cell. It's cortical epithelial cells. Then macrophages, antigen presenting cells. Epithelial, antigen presenting cells. Then you can see there's also nacelle. Nacelle is supposed to give nutrients. It's concentrated nutrients for the thymocytes. So that is the whole structure. And then another thing we should remember is that at embryonic stage, it is the yolk sac, which is hematopoietic, is the lymphoid organ. As development occurs, it changes to liver, then spleen takes over, then finally it's the bone marrow that now produces in a well-developed individual now. So let's look at secondary lymphoid organs. At this place we say the immune cells are sensitized. They are activated by exposure to antigen. Once that happens, clones of the same cell which have been exposed to the same antigen are produced. That cell proliferates. It divides to produce a copy of itself. Many cells which have the same antigen receptors. Then the other thing is that after activation, it is in these sites where they carry out the activity, mainly. So secondary lymphoid organs include lymph nodes, the spleen, and diffuse lymphoid cells. Diffuse lymphoid cells. So these are individual cells which are able to allow development and activation of the developing B cells or T cells. So they are called mucosal associated lymphoid organs. That means they are found on the walls of just below the mucous membrane. In gut, they are called gut associated lymphoid tissues. So these are another group of secondary lymphoid organ. The tonsillar lymph, the tonsil bronchial lymph node, and the bronchial or airwaves or respiratory tract. So let's start by looking at the first one, the spleen. The spleen is a very big organ. Let's start by looking at it. Spleen is a very big organ. It has capsule outside. Then they are trabecular, dividing it into regions or compartments. So in each compartment, you can see the red part. That is referred to as red pulp. It has also blood sinuses which supply the blood. So the red part is mainly composed of red blood cells. Then we have the vein, which is blue, and the artery, which is pinkish. So around the artery, there's this region which is blue and white. This is a region where white blood cells assemble. It is not a permanent structure. That depends on the white blood cells which leave the artery into there. So they are B and T cells. So this primary follicle, this is one focal region of very mature B cells. Then germinal center is composed of B cells which have been activated. They are called secondary lymphoid follicle, and now they are proliferating inside there. Then we have marginal zone, which I think composed of macrophages and the rest. Then we have periarterial lymphoid septum just around there. It contains those white blood cells, all of them. So that is the white pulp. So the white pulps are region of B and T lymphocytes which are developing. So let's look at how they develop here. How they develop. I've described the structure, so that is not very important. So red pulp, what happens in red pulp? Red pulp is just a region where macrophages now do not do antigen presentation, but they do kill the old and ineffective red blood cells. In the white pulp, this is where B and T cells continue developing, and they are activated by antigens. We have macrophage and electric cell presenting antigens to them, and then they multiply, proliferation, and then they also destroy what? They also destroy antigens. Also destroy antigen. So you can go through the function. Spleen acts as a filter to destroy antigens. White bulb site for BNT activation. Red bulb destruction of red blood cells. It stores leukocytes and platelets, help fight certain kind of bacteria. So then we move to the next ones, lymph nodes. Again, let's start by looking at the diagrams. The lymph nodes is also capsulated as a capsule with the divisions which can be seen. A unique thing about this is that it has something bringing in lymph fluid and carrying the lymph fluid away. So afferent lymphatic vessel brings lymph and efferent takes it away. So those are channels apart from artery and vein, which is there. So it is divided into outer cortex, the middle paracortex, and the inner medulla. So what really happens in these regions? Let's look at them. So the cortex is a region for development of B lymphocyte and its activation and proliferation. We've seen that. Paracortex is for T cells, activation and proliferation. Then medulla is a region where now they are escaping, but still there could be activation and proliferation. They are leaving the node. I've talked about the function of efferent and efferent lymphatic vessels. Then let's look at read through. This is what we have just said. They filter the lymph fluid, so they damage, they kill antigens, there's antigens, damaged cells and cancer cells. Help the immune system to build immunity by activating the T cells, produce and store lymphocytes under the immune system cells that destroy pathogens. Okay, so we'll see how these cells are stored there later. Then we go to the last bit, mucosal associated lymphoid organs. So these, I say, are aggregates of cells. They are not capsulated. They have no capsule, and they found in submucosa. That is like in the gut, they will find the wall of the gut, just below the mucosal membrane, the same to respiratory and urinary tract. So they can form nodules, aggregates, or they can remain as diffuse cells which are just collected there, but they do not form a body like a nodule. So they provide protection at the main entry into the body, nostril, respiratory tract, digestive tract, and urinary tract. These are regions where things can enter the body. So they also carry out surveillance. That means they walk around like police or security. And if there's danger, they get antigen, they clear it. So an example is in the gut. So in the gut, we have pious patches, which is the main malt or galt, it says galt. So it has cells known as M cells. These should be presenting antigens to the lymphocytes, B and T cells, mainly B cells. So once they present it to B cells, they are activated and they get into the lymphatics and move into blood and come back to the same wall and then secrete this antibody IgA that is transported into the lumen and kills microorganisms. Then finally, we have this diagram which try to show us the areas where this lymphoid organ is. Malt is found there, and under the anedoid, another one at the tonsils. Then that's thymus. Then we have the spleen there. Then we have pious patches, which is also galled, remember. Then appendix, malt, bone marrow. Then we have lymph nodes and lymphatics. Thank you until we meet again.